Good evening, everybody. We continue with the international Beyond Europe program, and the next session is focusing on the contribution of archaeobotany in connection to current knowledge on different crops that have fed human societies in different parts of the world. And I would like to invite my co-chair, Xingyi Liu from Washington University in St. Louis. He is associate professor there, the Department of Anthropology. I'm sorry for the long wait and that we are running almost an hour late in our program, so thank you for your patience. And I give the podium to Xingyi to start chairing the session.
Okay. Yes. Thank you, Tanya, and thank you very much for the organizers to put together a very interesting program. I learned a lot this early morning in St. Louis, in the United States. So the first talk we're going to have Professor Alex Levada from Catalan Institute for Classical Archaeology. Alex is going to present a paper which is the result of many co-authors, but Alex will be the presenter, which is entitled "A New Approach to Combining 3D Geometric Modern Morphometric Analysis of Machine Learning for the Identification of Past Land Races." Alex is archaeologist trained in the UK, but working in a variety of different sites in Greece. Also, the chief editor of Environmental Archaeology journal. So I will pass it on for Alex.
Hello, Xingyi. Hello, Tanya.
Hi. Hi. Good to see you.
Good to see you all.
Hello.
I'll share my screen. So thank you very much for the invitation and the opportunity to present the first results of our project that started actually in 2020 with the pandemic. So this is part of a bigger project that investigates the Aegean Early Iron Age. And as part of this bigger project, we aim to create new methodological tools for the study of ancient grains and agriculture. So, one of our research questions was how can we better use seeds and grains to make the most out of them? And our colleagues, our zooarchaeologist colleagues, from just a single bone, they can get so much information on pathology, on the sex of the animals, on cooking practices, and so much more. Whereas with seeds, we are rather more restricted, although indeed great advances have been made in the discipline. So our ambitious end goal is to use the full 3D morphology of ancient grains and link it to specific agricultural regimes. Also, we are aiming to use the 3D morphology of grains to potentially identify past land races. And this is very important to get insights into past biodiversity, especially now in the modern period with the increasing loss of biodiversity and with modern cultivation being based on genetically uniform cultivars, as we have heard during these 2 days of very interesting conference. So today, we will focus on this last aspect, on the identification of past land races in archaeobotany. And there are, of course, already different methods that are being applied to this end, and quite successfully. Although what is clear is that this is a huge field, and we're only at the beginning with much more work needed. So current methodologies include, for example, genetic studies and two-dimensional geometric modern morphometrics. In archaeobotany, over the last decades, geometric modern morphometrics are increasingly being employed to to improve information, for instance, on taxonomic identification, and also on the identification of variation within a population or past land races. Most studies to date focus on fruits, such as grape, olive, date, cherries, and this is because of their importance in certain past societies and the fact that are common finds in certain geographical areas. Less work has been conducted on cereals, although important publications are out with very significant results. So our work is an attempt to build on this line of research and improve our understanding of past land races of cereals and specifically of barley. We chose barley as this is one of the most common archaeobotanical finds across the Aegean, but also across Europe in all periods. Now, the premise of the new tool we are creating is based on the principle that shape encloses important information. So this has been observed by multiple studies on modern grains, but did not result in further investigations focusing specifically on their 3D morphology. And this is normal, is to be expected up to a point, because until recently, we had only very few and very expensive methods to actually reconstruct accurately in 3D the grains. So this is what, despite the challenges, we set up to do. And the first step was to obtain material. And to do this, we set up a collaboration between the Catalan Institute of Classical Archaeology, the Institute of Plant Breeding and Genetic Resources, the ELGO Dimitra, and the Department of Agriculture of the International Hellenic University. And through this collaboration, we set up a rather large area in Thermi, Thessaloniki, at ELGO Dimitra, for experimental cultivations. We chose 21 land races and 1 commercial cultivar. We included material from across the Aegean. We included land races from Crete, from Heraklion, Lassithi, from Santorini, but from across the Aegean, as you can see on the left-hand side on the list, Castoria, Leros, Drama, Florina, and so on. For our experiment, the design of our experiment was a split plot design with irrigation level as our main plot. The manure level was a subplot to this, and barley accession as sub-subplot factors with 3 replications. Our PhD candidate, Alexandra Kriti, who is at the audience today amongst you, is in fact actively involved in this part of the project, and she is in charge of the maintenance, monitoring, and the field work on the experimental cultivations. And thanks to the work of all the people at Thermi and Alexandra's dedication, we already have 1 successful year of cultivation and harvest. We harvested the material in the summer of 2021, and this has ensured the availability of material to move on to the next step of our the creation of our new tool. At the moment, we are in the 2nd year of cultivation, and we are now entering a very busy period of field tending with harvest that is expecting to take place relatively soon. Apart from the cultivation in various agricultural regimes, which we do to also control the impact of different variables on the shape and size of the grains, we took a series of measurements, many of which will be also extremely useful for informing on agronomic aspects of the different varieties. Apart from monitoring weather conditions, we monitor moisture levels, the plant height, lodging, chlorophyll levels, heading date, and so on. So after the first successful year of harvest, we collected our ears And we now have thousands upon thousands of grains from the different land races cultivated under different regimes. Now, with this material, we were able to pass, as I mentioned, to the second stage in the development of this new methodology. But because the material is huge, we had to start from somewhere. So we started with a pilot study using 4 land races, these of Lassithi, Chalcidisi, Larissa, and Santorini. We collected grains from the middle part of the ears, excluding seeds from the very top or the very base of the ear. To scan the seeds, we used an AutoScan Inspect Shining 3D scanner, and this scanner is a metrological scanner. And this is very important because it gives us real measures. It gives us measures of the grain directly without first having to go through photography and photogrammetry. So we can compare like with like, and we have achieved a very good accuracy of about 10 microns. The way to scan, we take 2 separate recordings of the grains, 2 recordings in order to ensure that we do not lose any part of the grain and the whole surface is scanned. And then for each grain, we scan it 3 times. And with this, we have managed to increase the accuracy. And also, this is done for quality control to ensure that there is no error introduced by the scanning process or by the different operators. So, with the scanned grain, we extract the landmarks. What are the landmarks? These are on every grain we have an irregular triangulated network, which is made of hundreds and hundreds of points, regularly distributed on the surface of the grain. Every corner of the triangles is a landmark, and these define the shape of a grain. Using this system, we transform the 3D object into a set of measures. And these are examples of automatically extracted simple measurements, which include sections at specific points and heights of a grain. These are just some examples of measurements, most of which are very difficult to obtain using traditional 2D geometric morphometrics. We are also including several indices and ratios that are developed by our team at ICAT that can provide very high discrimination for grains. So these measurements, instead of being analyzed using multivariate statistics, are instead introduced into a machine learning environment. Now, we chose this because we believe that machine learning has several key advantages. It uses already classified training data and can also test the accuracy of the method. So in other words, to explain this, let's say that we have 50 scanned grains with their measurements. We can use 30 of these grains, the measurements of the 30 grains, to train our algorithm. And then we use the measurements of the other 20 grains to test if our algorithm can correctly classify these grains to their landrace. The important thing is that machine learning does not take decisions on which measurements are most significant. It just uses all of them. And this way we avoid the factor of subjectivity. So this is our algorithm. This is how our algorithm, which is developed by Hector Orengo and his computational archaeology team, works. So here we import the training data. And this is an example of our training data. This is an example of 25 different measurements, for example, the height, the length of the grain, and so on. Every number refers to a type of measurements, and the class refers to the landrace. So this top line here is the header, if you like. So for each scanned grain, we enter their measurements and indicate their landrace. Every line is a record, and with these data, our algorithm is being trained. Then we enter the measurements here of a grain without indicating its landrace, and we ask the algorithm to classify it according to the training received. So this way, we can predict the landrace of a grain using the values of its measurements and then test the accuracy of our algorithm. In this case, by entering the measurements of a grain, the grain was classified as belonging to landrace 2. And this allows us to enter unknown grains and find the closest match. So far, we managed to identify landraces by 67% accuracy. This is good, but it's still relatively low. In our case, our ultimate aim is to identify different landraces with a minimum of 85% accuracy. And we are optimistic that this is achievable. This is very achievable because we just started the scanning process and we have a lot more grains that needs to be scanned. And the more training data you add to the algorithm, the more we will increase the base of the knowledge of the algorithm and its accuracy. Our computational team is also has a backup plan to use deep learning, which is a more sophisticated way in machine learning to improve accuracy. And of course, we need to take into account the effect of charring onto grains, because finally, we are creating this tool to apply it to archaeological material, and the archaeological material that survives is carbonized. And Alexandra Kriti will be leading the charring experiments, and the information, the data that we will collect from this part of the project will be introduced into the algorithm to account for this potential bias. And finally, of course, we have already secured several archaeological assemblages to try this tool. So, to conclude, what we have been doing, we are moving towards the 3D recording of grains to take advantage of their full shape, because we do believe that the full shape encloses important information, and by including it, we increase our base of knowledge. We are also changing the analysis methods, moving from statistics to machine learning, that we also believe gives us a great advantage. And with using the full complex morphology of ancient grains, we hope that we will be able to directly identify detailed human management practices and get insights into past biodiversity with very promising results to date. At the same time, the collection of the experimentally cultivated material gives us a great wealth of information to inform on modern biodiversity, on the quality of grains in relation to environments and agricultural practices, on the nutritional potential of the grains. And we think this will have significant impact on modern agronomic studies. Thank you very much.
Thank you so much, Alex, for a wonderful talk and new approach. I'm excited, and I can see the potential both in archaeological and modern research. Tanya, are we going to have questions now, or are we going to leave the questions at the end?
We will leave the questions for the end, and we will follow the program. Thanks, Alex, for this really exciting, novel approach. Our work on grave pips showed great variability, so for the Neolithic and the Bronze Age. So it's as if land races, varieties of grapevines were not yet there. So it would be interesting to see from your work what is the case with cereals, which will be great. Now we move to our next speaker, Professor Christine Hastorf, who has been working in archaeobotany for decades and has pioneered research in the study of ancient food. She is a professor at the Department of Anthropology at Berkeley University, at the University of California, Berkeley, and she is in the field in Bolivia, so she has sent us her recorded presentation, and I'm really grateful to her for accepting our invitation and for preparing this presentation.
Thank you for letting me speak to you today and record it. My name is Christine Hastorf, and when you have your meeting, I'm going to be in the field in Bolivia, so will not be able to attend. I begin with the Larson et al.'s very famous graphic showing 5 locations in the Americas for plant food domestication, noted across these 2 continents. Some regions actually have more than one area, especially South America. Here we see the origin of commonly grown vegetables, noting the globalization of food where plants are grown virtually everywhere. We're domesticated in many places and have moved around throughout the world, which is quite startling. So I wanted to talk about the land races that we find here in the Americas. And this was all done by the Columbian exchange of many, many foods that you are now commonly familiar with that have come from the Americas. And I just want to point out chili peppers, cacao, peanuts, avocados, tomatoes, as well as the potatoes. The American foods are surprisingly diverse. And these I chose because of their colorful imagery. I included a few animals in there too. So when you study the styles of development and transmission in the Americas, in these sort of 3 locations, you see that some plants were domesticated in one location, and we know this both from morphology and genetics, and they were exchanged across the continents. And the examples that stand out to me are maize and sweet potatoes. Some plants were domesticated in several locations and also spread out and then hybridized between themselves. And 2 that stand out to me are the chili peppers or capsicum and the beans, the Phaseolus vulgaris and lunatus. And some plants we're still learning about if they were domesticated in only one place or more, and those are the squashes and the sunflowers. So there's still a lot going on in this region. But what we do know is that the plants moved across these continents before Columbus. So just starting from the north and ending south, most North American plant foods were not domesticated, with the few that I mentioned, squash and sunflower and zizania, wild rice, being the most prominent ones that spread throughout much of the continent, this continent. Some, a group of them that you see listed here, have gone extinct, meaning there's only wild, sort of feral, I should say, examples of these growing, but also we find them in the archaeological record. And I know geneticists are working on bringing these back to make them more productive. And then when we turn to Mesoamerica or Central America, there is at least one center of domestication that you see here in green in that area, sort of the River Balsas region of the Guerrero state, which is sort of the mountainous region on the western coast. And we know that maize was domesticated there. Chili peppers could have come from that mountainous region as well. And then we also have amaranthus and Chenopodium nuttuli that also come from that area. And recently I've been working with Araceli on the chili peppers, and we believe that they could have even been domesticated over in this Yucatan Peninsula. So there's still a lot going on there. Moving to the South American group that you see here, of course, these regions are large and there are multiple locations. And there's many different food types that we know, but they also cover both warm and cool locations. So the taxa that we're aware of from these sort of three zones that were in the previous map are the beans, squash, and sweet potato. We believe at least some of them were domesticated in that northwest Amazonian area. The Andes Mountains have many taxa, the potato, solanum, which we'll talk about, quinoa, and other tubers, and lupin. Lupin was also domesticated. And in that southwestern Amazonian terra firma up against the Andean mountain, we know that also there were other beans, manioc, and capsicum peppers that were also domesticated there. So turning to this Andean region, this highland region, that long stretch, that long purple stretch on the original map, I just wanted to show you, especially those of you who aren't familiar with the American indigenous taxa, the range of them, that when I mentioned other tubers, there is quite a few other tubers that were involved in this region that were domesticated and engaged with, and still are. We just don't, they just haven't gone global yet. So when we turn to the potato, which is one of the two taxa I really want to focus on, this has had a lot of really wonderful studies of land races there. The wild solanum, we know botanically grow from the southern US, the northern North America, all the way to the farthest south, South America. So the wild solanum is everywhere. But the domesticated and selected species come from southern Colombia to the northern Chile and Argentine area. So it really is what we would call the heartland of the Andes, where the potato was domesticated. And there's two domesticates that we have. They're both Solanum tuberosum, and one is Andigena and one is tuberosum. And here's just a really nice picture of some of the highland, the Andigenum examples of varieties from Central Peruvian Andes. These are all the diverse land races. And the diversity is a response to environmental conditions, but also the other things that have influenced potato, which is day length, frost, days of frost, and also most primarily culinary flavors and textures that the farmers particularly like. The number of land races vary across the region, of course, but there's three main groups that my colleagues Steph DeHaan and Rodriguez have focused on in their research over the last 20 years in the Andean region. And they call them commercial flowery land races, and those are the ones that you get the big, the larger specimens of. The non-commercial flowery races, which is what you were seeing in the previous photograph, and then the bitter land races. And these are, most of these land races, not the commercial ones, but the other two, are managed and grown by smallholder farmers in the Andes, in the Chilos Islands, for that region. And I just wanted to highlight something about the bitter potatoes, which is a distinctly high crop, high Andean crop, and that the southern Titicaca Basin, where I work, is one of the hot spots for this, to show you about this. And what makes this unique is they're freeze-dried in the winter, their winter, which is coming up July, August, and they do this to, one, get rid of some of the chemicals that are make them inedible as they are straight. You can't just boil them straight. And the other is to freeze them and be able to therefore freeze dry them and be able to keep them for years. So you can really, this does much work, this processing for this potato type that is basically not edible. This land race is not edible, just boiling straight. And this study by Brush and his colleagues really stands out in the study of land races to give you a sense of how these, how diverse a farmer's field can be. They made these symbols of these different types. These are Quechua terms. This comes from a field. They plotted a field in Chinchero, which is near Cusco. And you can see how each individual plant really grows next to a different landrace. So it's really quite diverse, up to 90 specimens, 90 varieties or 90 landraces within one field. And I also wanted to bring in a bit about Bolivia, that up to 70% of all potatoes grown in the Bolivian highlands are landraces. So turning now to chenopods, the second of two taxa I want to mention. Chenopodium is a wonderful taxon which I think we will find across the globe, grown across the globe and harvested across the globe. But in the Americas, there's three distinct independent domestication processes, one in Mexico, one in Eastern North America, in that blue spot I showed you on that map before, and then in the Western Andes. And I'm going to focus again on the Andes example. And we know of today from research by a series of agronomists and scholars, archaeobotanists, that there are two Andean Chenopodium domesticates. You know the Chenopodium quinua, that first name on this list, but there are two accompanying weedy species that are often found around the fields, and they're listed here. Melanospermum is the more common. It's called quinoa negra, black. And then the two wild species, the Ambrosoides and Hercynium, those are both also, like the weedy species and the domesticate, harvested and eaten by highland people. So there clearly is a lot of encouragement of all of these 6 taxa. We have data on the chenopods from a series of locations, but I'm going to focus on the area I work in, and my colleague Maria Bruno has done this good work. I'm going to see if I can move me down a little bit so you can see that. So obviously farmers grow this throughout the Andes and now throughout the world. It's shipped out from the highland Andean countries to the world now. But the other, that's the quinoa, but the other is less well known and is not global, and that is the quinopod paleocaule, also called canyawa. As well as, and it is unique because it is self-seeding and has different maturation rates and also is easily shattering, much like the weedy species that we know. So it still has a different sort of landrace group of characteristics, which probably makes it not going global, and the yields, of course, are slightly less. So to identify this archaeologically, we look at a series of attributes, as all archaeobotanists do, on seed study, and we take a series of measurements. We look at the diameter, the testa thickness, the texture, which you see here in the drawings on the right, and also importantly, margin configuration seen on the left, and beak prominence. That's a new trait that we're finding very, very useful that we've been working on. So an example, a final example, is a landrace study, an archaeological landrace study that we've completed here in this northern Bolivian area. Maria Bruno worked on the Chiripa remains, and Brianna Langley, Maria, and myself have worked on the area to the south called La Barca. And we were comparing all these attributes to each other and to the modern varieties. From this work and others, we know that at least by 1300 BCE, domesticates and sort of intensive farming of these plants have been going on. So here are the data very briefly, and I've tried to write the names of these taxa in their clusters of symbols from our publication. And you can see that Chenopodium quinua in the lower right is very unique and separate. The Chenopodium melospermum, a weedy species, is also quite separate, but then you get a cluster of the wild Ambrosoides, the canyawa, as well as the archaeological seeds from La Barca. So that gives us some suggestions that the La Barca seeds are the progenitors of perhaps the canyawa, or in fact even perhaps a different variety. So the archaeological kenopods do display a unique mix of wild and domestic attributes, suggesting that selective process of these land races that are going on. Now, the Labarca seeds are not found today. While they're close, they're not the same. So again, we see these sort of varietal land races continuing to be grown in this region and were grown in the past. So, but also, we don't find the Labarca seed up in that Charipa area, suggesting that group in the south were selecting slightly different land races than the group around the lakeshore that we are finding at Charipa, also suggesting quite a lot of varietal difference in the kenopods on the land. So with that very brief tour of land races and taxa throughout the Americas, I'd like to thank all of you for listening to this, and thank you, Tanya, for including me. And I'm really sorry to miss the whole presentation, but thank you very much for being here.
Many thanks to Christine. Thank you, Christine, for this fascinating presentation. You managed in 15 minutes to give us an overview of the prehistory of these plants and connecting the points you made to the discussion we had yesterday in the round table. It's interesting that we were discussing whether a local variety of aubergines cultivated in a Greek island the seed can move to different places. But we see from your presentation how quinoa has been spread all over the world, and instead of indigenous people making a living from their own seed, now the seed is sold, is cultivated under large scale. So it seems like there are different lines of how things work. The micro scale of an Aegean island and its seeds, and the large globalized market that operates on a different scale. And the important aspect, I think, of Christine's contribution is that she has shown us how land races are very closely linked not only to the environment, but also to the taste that people have and the foods they cook with these plants. So there is an interaction. Now, Shingi will continue chairing the session.
Okay. Yeah, thank you very much for Christine. That's a wonderful talk. So with our next presentation, we'll move from the New World back to the Old World, back to Caucasus. So we have Dr. David Margherita from Caucasus International University in Georgia, where he's the head of the department at the Institute of Horticulture, Viticulture, and Oenology. And he's going to have an active program in research program in Caucasus, and he's going to talk about grape wines in Georgia from Caucasus. And I can share your screen.
Thank you for my presentation. I thank you for organizing to give me opportunity to present our work, which is dedicated to the Georgian local varieties. And I would like to first present to you some information about Georgia and its viticultural region. We are a country located east of Black Sea region. We have 12 wine regions inside the country, in all parts of the country, east as well as about 5,000 hectares of vineyard are in Georgia recently, and producing something 250,000, 270,000 also of grapes. Among those, 55% are white wines and 45% are red wines, which is very important. Georgian local varieties are cultivated in majority in our country. We can say that 95% of Georgian vineyards we have a cultivation of Georgian local varieties. And each region, each viticultural region is famous with their local varieties. But among those, Cacetelli Sapera, we are well-known varieties. Mazeli are leader varieties inside the countries, as well outside also of Georgia. We have 24 appellation of origin wines zones, and our wine export is in progress. But today we should focus our presentation on diversity of Georgian grape varieties. This small country is very rich with indigenous grape variety. We count 525 indigenous grape varieties, and which is confirmed by ampelography DNA markers. But those are originated in different parts of the country, having different characteristics. Among those, 437 unique grape varieties present in our collections, but they are not only just subject of preservation, they are also for cultivation.
Professor, sorry to interrupt you. I don't know if you are changing the slides.
Yes.
We don't, okay. So I just wanted to check in case you had it on, we did not see any change in the slides, so.
Now?
Now, yes. Thank you.
Okay, so another important milestone for Georgian varieties is that they are subject of different breeding program. It's inside the country, inside Georgia, outside also in different breeding programs. In total, if we summarize, 193 varieties was bred in 21 countries with participation of 13 Georgian leader cultivars. We speak about high breeding potentiality of our genetic resources. Now, looking for origin Georgian local varieties and history, we are going very deeply because the Caucasus region, South Caucasus region is recognized to be the center of wine origin, grape domestications since long time. On the slides, you can see several different visual presentation of the maps, and the list of notable researchers mentioned and noted about importance of South Caucasus in grape wine origin and wine domestication. The last confirmation we have from the McGovern and co-authors, which published their work, Early Neolithic Wine of Georgia in South Caucasus in 2017 in the Nation Proceeding of National Academy of Science of the United States of America, in which it was multidisciplinary study given new information about presence of organic acids, including tartaric acid, as a marker for wine, vitis vinifera wines, and also it was demonstrated that grapevine grows close to the village. This means that the people, in Neolithic people, they have also initial step of viticulture as minimum. To have domestication of grape wine, we need to have crop wild relative, wild ancestor, which is, in case of cultivated grape wine, vitis vinifera silvestris. It is typical representatives of Caucasus and Georgian flora among those. It grows in woody region, forest, lowland, river banks up to 1,002 meters above sea level. Its population is decreasing due to phylloxera, downy powdery mildew infection, but even today we are able to collect 75 genotypes wild grape in field collection, and we did ampelographic description using OIV descriptors during 2018-2022, which demonstrate large diversity of these genetic resources. And probably this was the initial step for domestication, also our cultivated grapevine gene pools.
Please check the presentation again, if the slide changed.
Now, it's okay?
Now it's okay, yeah.
Okay,
it's okay. Yeah.
So, okay, I will do it this way. So, as you can see, another approach was archaeological-botanical research with object to utilization geometric morphometric to characterize ampelographic grape seeds in comparison to modern wild and domesticated grape sound from the Caucasus, another part of Europe and Mediterranean world. There was include 450 British archaeological seeds from 40 sites in various locations of Georgia since early Bronze Age until the 19th centuries, and elliptic Fourier transform analysis was used for shape study by Laurent Boubi, University of Montpellier, France. And this research demonstrates that ancient samples we have not very few, they are only few, but they are mostly domesticated, the wild types, domesticated morphotypes identified, and usually they are predominated from Iron Age and onwards, and strong diversity of domesticated shapes close to modern cultivars from the Caucasus and Greece area became evident after this study. Another party was to start ancient DNA of archaeological samples. Here we had also 21 archaeological wiki seeds, and it was used, DNA isolation was covered by Illumina libraries for next generation sequences. Shotgun sequences was done. The time period was from 1000 BC to up to 18th century. And during this study, it was just 4 successful accession from Borjomi to Santauro and Tsamuri, and they are linked to modern Georgian varieties like Asuretuli, Shawi, Adreoli Skeltana, which demonstrates that there are some roots in modern genetic resources of grapevine coming from ancient, as minimum, medieval period. Another study of genetic resources grapevine is dedicated to ampelographic study, because to make characterization of varieties, which started since 2003, and we have quite good evidence of diversity of different ampelographic traits, like color of berry skin, the ancient berry size, and sugar contents. It's also demonstrated that Georgian local varieties, they have late ripening patterns respect to European varieties, which is very positive for, in the case, modern climate change situation. Last decade, very important, significant work was done for research resistance to downy mildew of grapevine, and which is very important disease and very significant to discover genotypes which can be some level of resistance against this pathogen. So it was selected 6 varieties with more high resistance, and among those, Galoblishvili became very important, because in Galoblishvili it was identified 3 high significant novel loci against Plasmopora viticola named 21 to 3031, because they associated low level of pathogens correlation, conferring from high to very high resistance to downy mildew. Because why it is so important? Because this is first evidence of resistant loci against Plasmopora viticola inside Vitis vinifera germplasm. Because before, all genes they were defined, identified in American or Asian grapevine gene pools, which have the low quality of grapes. This makes new gate to the future breeding programs. Another new, very pleasant news is that 2 Georgian varieties, Shaltsinska and Svedian, etc., they also demonstrated resistance against another very significant fungal disease, powdery mildew. And so they are important also to be included in the breeding programs in future as for cultivation. Another important, very important news is the resistance of grapevine yellows in Georgia, because they demonstrated severity symptoms against this pathogen. Infected Georgian varieties are infected, but majority of this variety showed moderate and mild symptoms, whereas the international cultivars exhibited severe symptoms. Range of symptoms, intensity suggests different susceptibility of such cultivars to both noir grapevine yellows in Georgia. DNA study, which was part of this investigation, Georgian local varieties, demonstrated geographical linkage of varieties to west and east part of Georgia. This was demonstrated by SSR markers and by ethnic markers as well. They are very well concentrated to geography or inside the country, but as well because we have different climate reality and soil condition in west part of Georgia and east part of Georgia as well. So both techniques, like SSR markers or SNPs, confirm that Georgian genetic resources are very well structured into very important clusters, and they are also linked to Vitis silvestris of the country. Very important is to see how Georgian genetic resources are related to another gene pools. Here we have a result based on the macrosatellite marker as a SNP markers. Here in the right, you can see the Georgian local varieties with blue dots, and in comparison with French, Italian, Spanish varieties. And you can see those are very distinct from these genetic resources. As well, they are also, despite there are some genotypes which share some others with West Eastern European varieties, Georgia makes separate cluster, which are, it seems that it's new genes not involved in breeding East and West European varieties. So this can be some perspective for future program as well, breeding program of cultivation. Very important is also in study of grape genetic resources to make characterization of genetic resources. Since 2012, we did some progress for characterization, description, Georgian local varieties, as well as some wild grapevine accession of Georgia, because to see a relationship from wild to cultivated one. So these are very important also for preservation and conservation of these genetic resources inside the country. Today there are several collections having more than 3,000 accession of grapevine originated from Georgia as well from neighbor countries as well. And all this work became possible in cooperation of Georgian institutions, universities, research foundations, and also universities and researchers from different parts of the world interested about grapevine genetic resources of Georgia. We are very thankful all this institution having attention and interest to Georgian local genetic resources of grapevine. And thank you for your attention. It was a very pleasure to present you a result of our work for last year.
Thank you so much, David, for such an informative presentation on a very important issue of wine domestication with the Council and the true results of international collaboration. So I will hand over the torch back to you, Tanya.
Thank you, Zindy. This is really fascinating talk we listened to, and hopefully we can discuss a little bit later. And now our co-chair, Zindy Liu, who has been very kind to accept the invitation and travel us further eastwards to the places where rice and millet were originally brought to cultivation and domestication. Zindy is a colleague from the Washington University in St. Louis, so look forward to your talk, Zindy. Thank you.
Thank you. Let me share my screen. You can see my screen, okay? Yeah. Okay. Okay, thank you very much, Tanya, for that generous introduction. So what I'm going to do in the next few minutes, I want to follow Professor Christine Haftov's steps to give you a very brief introduction, very brief summary of the changing crop choices in East Asia, particularly in China, focus on millet and rice land races. So this is a summary of my talk. I'm going to particularly highlights some of the archaeological features in relation to human-environment landscape, human-environment relationships. So I want to emphasize the human dimension, the social dimension of food choices in the past 10,000 years. And I'm going to focus on 3 episodes of the development, the early Holocene, mid-Holocene, and in each of the episodes, I'm going to highlight particularly archaeological features, starting with the human landscape, and then I will unpack the process of a cross-continental movement and the role of that movement plays in the biological domestication, and the talk by highlighting some of the key features of the culture choices of a crop, changing crop choices. One of the interesting features of the origin of food production in East Asia, there is a sort of a underlying common geography. This is one of the map I found on internet, but I think is very unique. Give us a unique perspective to understand the topography underlying early agriculture in China, in East Asia, between 10,000 and 7,000, roughly 10,000, 7,000 years ago. The great thing about this map, you see the topography, you see the uplift of the Himalayas, the Tibetan Plateau, the Tian Shan, Himalayas, and Kunlun, so on and so forth in the far distance. And then you have this second tier difficult terrains, including the Lhasa Plateau, Gobi Desert, and the Mongolian Desert, as well as the conjunction between the Lhasa Plateau and alluvial plains of a series of big rivers such as the Yellow River Valley and the Yangtze River Valley. And this is one of the first academic paper I published a while ago, highlighting there is a common geography, there's a common topography underlying the early key site. With the last 20 years of development of archaeobotany, we have a much better understanding of the domestication process of, for example, rice in the south and millet in the north. M represent millet, broomcorn, foxtail millets equals rice, domesticated rice. And as you can see, those sites are not located in direct association with the big river valleys, as we previously assumed in the discussion of origin of agriculture in East Asia, but rather the sites are all a series of key early farming sites are located in between ecotones, between the Lhasa Plateau, at the foothill locations hugging a series of low mountains bordering the Lhasa Plateau. So in one of my papers are highlighting there's a, so if you think about the topographical conditions of early farming, so there is eastern hilly flanks. The geography very much resonates with the hilly flanks of the Fertile Crescent, where the Fertile Crescent founder crops was domesticated in Southwest Asia. Here in early Holocene in East Asia, you have rice and millet was domesticated in a similar topographic context. I think there are two things important for those locations. On the one hand, we know those archaeology, we know those prehistoric communities share a certain degree of a common membership in typology. They use the same type of potteries and stone tools and lithics, so on and so forth. At the same time, they are managing the same type of ecological niche. So this is slide zooming in a little bit to look at some of the key archaeological cultures that provide early evidence of food production. Xinlongwai in the north, the Peiligang groups in Henan province, for example, the Holy groups in Shandong Peninsula, and the famous early right sites at Xianyandong and the Diaotonghuan in the early Holocene in Jiangsu. So as you can see, they all share a common topography in between ecotones and hugging the mountains located at the foothills, rather than we previously assumed the early agriculture started from the river valley. The other thing these sites are sharing in common is, although rice and millet domestication has been the focus of discussion in the last decade also, but in fact, the archaeobotanical data suggesting rice and millet is only a small proportion, of small component of a much broader food choices in those diverse eco niches. So the first episode, roughly between 10,000 and 7,000 years ago, we see local choices. So eventually, crops as a component of the food web in rain-fed food locations or between ecotones. And next, I'm going to move to the next episode to see some of the features in between. And this is the classic East Asian Neolithic period, so roughly between 7,000 years ago and 4,000 years ago. And during this time period, we see two processes that happen roughly at the same time. On the one hand, we see a much global process of a transuration movement of the domesticers. And this is a sort of a collective work as a result of my own research as well as a lot of contribution of colleagues around the world. I think one of the interesting development in Old World archaeology is this realization of the transuration movement of cottages and livestock, and particularly crops, in long distance, in continental scale, way before 1,000 years before the Silk Route. So a different sector of the Old World was well connected by agriculture technologies way before. The process starts roughly around 7,000 years ago, and by the second millennium BC, we see on the one hand the Fertile Crescent founder crops, notably domesticated wheat, travel all the way from the Fertile Crescent to East Asia, across the Iranian Plateau and Tibetan Plateau, introduced, and you see a lot of them in the archaeological records in the Yellow River and eastern China. And you have the Chinese millet, Brunquan, the foxglove millet, more or less follow the same steps, the inner Asian mountain passes and get to Caucasus, and by the second of millennium BC, for example, 10 years of work shows millet was documented in archaeological assemblies in Greece, in Thessaloniki region. In the middle of the second millennium BC, they also carbon dated to about 1500 calibrated BC in Eastern and Central Europe. And this is also the time periods right of moving in long distance across East and South Asia. There's also interesting parallel movement between North Africa and India. So on the one hand, things are moving in long distance, and some of the tradition, and during that process, some of the traditional package, like the Fertile Crescent crop assemblies, was unpacked. And by the end of the process, they combine, they meet with another group of the crops, forming a very different crop assemblage with a mixture of newly introduced and indigenous crops. The other thing happened during this time period is we start to realize, and this is again through a number of excellent colleagues' work, such as Dorian Fuller, we start to realize the domestication process is a slow process. And this is one of the classic papers suggesting that on the right-hand side, on the left-hand side of the screen, you see the morphological phenotypic difference between the wild and the domesticated rice in terms of the shattering effect, the taphoretic internodes. So the undomesticated wild ancestors have taphoretic internodes, and the domesticated has a clean shattered grains. On the right-hand side, you see the chronology, and this is all from the same site, Tianluoshan in the lower Yangtze River region. You see the fixation of domestication traits, in this case, taphoretic internodes related to the reproductive isolation, was a slow process, not until the whole process started about 7,000 years ago, not until the Liangzhu period, 4,000 years ago, you see a dramatic shift towards a much higher proportion of domesticated types. So domestication of rice is a long-term process, and this is well documented in the lower Yangtze River Valley. Similarly, in the north, millet is very small, so it's very hard to extract the retic and chaff evidence from the archaeological assemblage. But nevertheless, for example, the work of my colleague Jimmy Zhao from Institute for Archaeology in Beijing document a gradual shift in grain sites, for example, in one of the key Neolithic sites, Yuhuazhai near Xi'an. So as you can see, from 7,000 years ago to about 5,000 years ago, and this is from the same archaeological complex, through different phases, the grain size of folk minute, Cetera Italica, increased gradually. We are writing a new paper suggesting at a sort of national level, at a global level, the grain size. There's some doubts about the grain size, using grain size as one of the domestication syndromes, because the data is primarily driven by regional diversity, regional variations, rather than diachronic shift from small. But nevertheless, you see a very slow process. So the interesting question, I always think the two processes are somehow conceptually connected. On the one hand, you have this early movement of domesticated and land races across continental at a continental scale between 7,000 and 4,000 years ago. And on the other hand, this is the key episode of a biological domestication, fixation of a key morphological traits such as a grain size and the shattering effect, which is well documented in millet. And this is one of the schema I borrowed from Martin Jones, my mentor back in Cambridge, who's really highlighting the conceptual connection of these two processes. And one way to think about it is in the core areas defined by assumed distribution of wild ancestors. It's very difficult to manage your newly domesticated land races because it's very difficult to block the gene cross, the integrations between the wild stands and the newly domesticated land races. And in the context of a trans-Eurasian movement, the earlier periods of domestication traits are often documented, and this is both the case of millets and rice, I think, beyond the assumed cause, with a reduction of environmental triggering, because the crops is facing entirely different environmental and cultural conditions. Therefore, it trigger further selections to adapt to the new environment, and also the improvement of reproductive isolation. And this is simply because the crops move out of the assumed distribution of a wild ancestor. And increase, I think we are in early stages of seeing this picture of sort of the globalization process play an important role in the process of domestication. And again, borrowing Dorian Fuller's work, on the one hand, we document a slow process of rice domestication in the lower Yangtze River Valley between roughly 8,000 and 4,000 years ago. But if you look at the distribution of the rice in this time period, rice have traveled all the way across East and Southeast Asia. It's hardly to not think that early movement of land races and newly domesticated crops may play a role in the understanding of the chronology and geography of the domestication process itself. So that's my second episode. I'm highlighting the potential, the possibility, the potential discussion of the cross-continental exchange and the role played in the biological domestication. And very quickly, moving to the culture choices. I think I missed one of them. One of the. I'm going to move back and forth a little bit. Sorry. Moving to one of the culture choices. So the interesting question is how food, and this is one of the key questions in archaeological research, how food production and subsistence may play a role connected with the social arrangement. One of the interesting features in Chinese ideology and cosmology, and this is pointed by the excellent agricultural historians Francisco Bray, who highlighting there is an exceptional feature in early China, which is the absence of rice deities in China. If you think rice play a very important role in the subsistence economy throughout the prehistory and the historical periods, but there is no rice deity. And this is one of the exceptions for Asia. If you look at other cultures in East and Southeast Asia, wherever you look, you have a very strong connection between rice cultivation and cosmology in Japan, in Southeast Asia, so on and so forth, where China is the exception. China don't really have a very strong tradition, but nevertheless, there is a millet tradition in China. So one of the potential answer to this unique feature is this is traced back to 221 BC, when China was first unified by the first Qin Empire. And the Qin state was an economy very much based on the millet cultivation in the Lhasa Plateau. So somehow that's millet-based ideology was able to conquer the China associated with the process of unification in 221 BC. So this leads to the question: what is the relationship of the social arrangement ideology on the one hand, and the infrastructure and the economy, subsistence economy on the other hand? I want to highlight that this research, which is quite interesting, I found, which is research done by psychologists and the sociologists, looking at different social and psychological arrangement between different parts of the China, where you have a very different underlying agricultural traditions. So they separate, really they're testing the rice paddy field hypothesis, the separating regions from the northern areas where you have a lot of wheat and millet cultivation today, from the green areas, as you can see, which are largely dominated by rice paddy field. The assumption is that because the people are cultivating different grains for thousands of years, this may somehow shape and articulate the social arrangement and the psychology associated with it. And they do found evidence, psychological and sociological evidence supporting their hypothesis. So basically, in the northern wheat and millet areas, people are more individualist and culture thoughts. One of the example is if A is true, it's very analytic, Western style analytic individualistic thinking. If A is true, then not A is false. In the paddy rice areas, the society is organized in an interdependent, holistic arrangement. One of the example in the psychological questionnaire, if both A and not A is true. So I'm not totally agree with it, but nevertheless, I find this a fascinating research. Sort of generates more questions rather than answering any of the questions. This is from the same research. They look at the marriage system of the two different regions. So it turns out that if you live in the communities associated with the paddy rice cultivation, the divorce rate as well as your psychology is different. So moving from northern China wheat and millet farming system to southern China rice farming system, you have two different distinct marriage system, and this has an effect of the divorce rate. Another following up research looking at, and this is fascinating, I want to share with you, I think it raises a lot of questions. They look at the way in which people sit together in coffee shops like Starbucks, which is a new phenomenon for China. And they found out that people organize themselves in different ways, north and south. In the wheat areas, people tend to sit alone, both in the weekends and during the weekdays. In the southern paddy rice communities, people tend to group themselves in coffee shops. And there's evidence that people tend to move the chairs to form a particular block, form a particular small-scale community so they can talk to each other, where in the north, people tend to sit alone. I think all these kind of raise a question rather than answer any of the question. But it's a meaningful question in the archaeological research. And it goes back all the way back to the original debate between Karl Marx and Immanuel Kant, whether it's the infrastructure dictating the superstructure or something. So eventually the question raised from this research is whether the crop choices, the choice of food production was some byproduct of a social arrangement or the other way around, or whether it's the case that the choice made by the early farmers domesticating different different species has a long-term consequence of a social arrangement in the political ideological spheres. So I think in many ways they raise the questions rather than answer any more questions. So very brief summaries. In this 15 minutes of talk, I go through some of the key episodes of changing choices and I highlight we see a picture of a changing from the local land races, where the crops choices and also the site location choices very much depends on the local environmental triggers, and the crops as a component of the broader food webs in rain-fed, food-hill locations, a situation resonates with the Fertile Crescent in Southwest Asia. And in the second episode, during the middle of the Holocene, we see two processes happen at the same time. On the one hand, domestic herds move in long distance across Eurasia, cross-continental scale. On the other hand, you have a very slow gradual process of fixation of domestication traits. I'm suggesting there's a conceptual connection between these two processes in the Neolithic. And the cultural land races really are highlighting that since about the fourth of the Bronze Age onwards, there's a very pronounced cultural choices made, influencing the way in which crop production and food production was organized. And the question to be raised there is whether the adaptation of land races to narrow cultural choices was the trigger, or the other round, or is it really the choice of the land races that lead to a certain social and political arrangement. So with that, I want to thank for your attention, and hopefully I managed to give you a very brief introduction of the entire Holocene in East Asia within 15 minutes. I want to thank some of my collaborators and my students, and also the funding bodies supporting my research in the past. Thank you very much.
Thank you very much, Xingyi, for this fascinating talk, and it's the perfect way to end today's session, because what follows is a round table, because you have managed to connect the environment, the land races, and culture. So it was taste that was brought up by Christine's talk, and with you it's how society is organized, social relationships that are behind the choices of land races and what people grow, which I think is, it brings us back to it's not only good to eat, but it's good to think about food. So thank you very much. I have been asked to that we don't have much time for discussion, so shall we say that we can have one question for each talk? And do you want to, so we will, yeah, you start with Alex. And would you like to chair the questions as we did the talks?
Yeah, absolutely. So we can we can entertain some more questions to the speakers of this talk.
Are there any questions in the chat?
I have a question to Alex, if Alex is around.
Yes, sure.
I can. Yeah, that was a fascinating talk. I'm very, very excited. I've seen some of the recent publications using micro CT. Have you considered the potential? And what's I'm not an expert on the 3D morphology, but in your view, what's the advantage and disadvantage of using different type of the methods, micro CT versus more sort of conventional 3D scanning?
I have to say that we have not considered using CT. I think partly is because it's a very expensive equipment and it's not available to everybody. So what we are trying to do is try to use commercial scanners. For instance, the scanner we're using is a typical scanner that all dentists can use in order to be able to make this tool available. And since at the moment we do get results using a simpler scanner, I think we will continue this way. But CT definitely offers different types of information. So yes, perhaps something to think about in the future.
Yeah, that's great. Yeah, I was also sorry for following up a question very quickly. I was also thinking about so in your samples you tested both the 2-row and the 6-row. So presumably the distinction of the side grains of the 6-row
are different.
Are very different, and
yes.
In some ways will drive the pattern.
Yes, yes, definitely. And even from the 6-row, we have 2-row grains and 6-row grains. So all this information is information that is entered into the algorithm to be taken into account. At the moment, because we are at the beginning of the scanning process, because we have to scan thousands and thousands of grains, we started only with the twisted grains. So everything we have done so far are on twisted grains.
Yeah, that's great.
But yes, it's a very good question because there are a lot of factors to think about on known morphology and other variables. But the good thing with machine learning is that we can enter, the more data we have, the better. So we just input huge amounts of data, anything we have there. And this helps us filter out biases.
Yeah, wonderful.
Okay. Να το αφήσουμε αυτό για το τέλος. Yeah. Alex, I have a question too. Wouldn't it have been easier if you had chosen the grain of einkorn, for example? Because with barley you have many more variables to play with, and whereas with einkorn it would have been more straightforward to develop the algorithm and then make it more complex?
It actually, yes, you are right. Barley is complex because also you have to think of another issue, the dehusking process, which is really, really very time consuming. So yes, the next stage in the project is to cultivate einkorn and free threshing wheat as well. And Yannis Milonas, we have been discussing this with Yannis Milonas and Lisa Ninu, and this is the next stage that we are hoping we will start growing grain from these wheats.
Then make it more complex?
It actually, yes, you're right. Barley is complex because also...
Shall we move to the next speaker, Jinyi?
Yeah, absolutely.
Rico, did you have a question for the first speaker? Okay, now we move to Georgia and the very interesting talk by Professor Makradze. Any questions for this talk?
Yes, I have one question. First of all, I mean, all the presentation was very impressive and very informative. And there's a very, very good base to think more about all the history of domestication. I'd like to ask what is the status of genetic erosion of vine cultivars in Georgia? Thanks.
So, as you mentioned, we have by listed 525 local varieties, indigenous varieties. But recently, in our collection, we have just 437 varieties. The epidemic of downy powdery mildew phylloxera, they started to reduce genetic resources in 19th century. In 20th century, varieties which was still available in that time has been collected in different collections. But as you saw, we still have missing varieties, which even according to lists, they are missing some dozen varieties which we have no anymore in our collection. And regarding also cultivation, 32, maybe 50 varieties is at the cultivation, and others are inside the collection. So we wish to have more varieties inside the cultivation. Recently, there are some trends because grape growers are looking for some rare indigenous variety, and they try to return back those to cultivation. But the process is slow, and we wish to have more varieties back to cultivation because we know that grape under cultivation is more preserved than it is inside the collection.
Thank you. I have a question that I would like to ask to you, Professor Makradze. It's really fascinating that you brought to us all this long-term presence of the grapevine and the variety, and it is considered one of the earliest places of grapevine domestication. As it is an agronomy conference, I would like to make the contribution from our project Plant Cult that ended a couple of months ago, and we analyzed hundreds of grape pips from the Neolithic to the end of the Bronze Age in the Aegean. So we have here on grape pips dated from 4300 BC, we have been able to see how the grape pips change from morphologically wild to morphologically domesticated. So there may be different areas, one of them being Georgia and another one being the Aegean, where people experimented with wild grapevines leading to domestication, and perhaps this might account for this DNA difference you see in your, if I understood correctly, in the Georgian varieties that you have that separate from in the diagrams you showed us. And I don't know if in the archaeological traditions of Europe, of Georgia, you have a wine god. In the Aegean, we do have, to connect it with what Jingyi said about the millet god in China. There is a wine god in the Aegean that is, he is as old as 1300 BC. So yes, this is, I wanted to present to you the data we have from our area. And would you like to share your thoughts on that, please?
Right. So yes, because by historical point of view, very often there was abandoned grape varieties. Then, because of existing wild grape inside the forest, it was gene flow probably between wild and cultivated one. It is common because also the gene pool of Georgian local varieties is dedicated by classification of Negru. They are Prolis pontica. So Prolis pontica is typical with small bunches, wine grape varieties. So, and also by historical point of view, it was noted abandonment of cultivated grape varieties, interaction with wild grape, and because of until mid of 19th century, before the infestation of phylloxera, downy powdery mildew, it was quite a large amount of wild grape living around the cultivated grapevine. So probably, with high probability, and it is documented, it was gene flow between cultivated and wild grapes. That's why, because of country in last period was how to say, autonomous developed, probably the genetic resources which we have today, and as they are distinct from East and West European genetic resources, is a result of local breeding programs, which has not too much interaction with another breeding programs of West East develops. Yes, we find some accession, some varieties which are linked with East, for example, Caucasian varieties, Iranian varieties. It's normal because these are countries to which Georgia had cooperation, exchange of trading, etc. So, but in a large sense, respect to Europe, this is evidence that they are different from European genetic resources. And of course, with Greece as well, because the Greece is Georgia, we had a long time of historical linkage. We have some genetic resources, some varieties which can be linked to each other. With Greek varieties, is a long history of, historical history.
Indeed, we have connections. My great-grandfather was from a village near Tbilisi, so...
Okay.
Thank you. And thank you very much. Any questions for Jingyi? Δεν θέλετε εσείς? Ναι. We have a question for you, Jingyi.
Yeah.
Okay. Thank you very much, Dr. Singli. I wonder how all these findings from historical data, and because you are an anthropologist, I know historical anthropology, all this, all these findings, how will affect your outlook on modern life, on today's world? Could you explain us how you feel? Because we have a lot of changes this season, climatic change, health, yes, and all these from resources. And please, let us know your opinion about this.
So yeah, I guess the question is how this is resonated with our experience of food and the choice of language in the modern context.
Yes.
I mean, I don't think I have a direct answer to that's a question. I'm in the early stage of thinking about these things. On the one hand, I think a lot of the things that shaping our own life is very much newly invented. And we tend to think there is a deeper tradition, but sometimes those tradition was newly invented, subject to the conditions when they were invented, probably during the last 200 years. So on one hand, I don't think we should read too much into this long tradition of sort of a deeper DNA of the cultural organism, if you like. On the other hand, I think there are common patterns. There are common patternings that we see in the cultural diversities in the past versus resonate with our experience. One of the things I've been thinking a lot in recent years is this key episodes of a movement, key episodes of globalization, if you like, in the Bronze Age, when things are moving in long distance, domesticated animals and plants move from one side of Eurasia to the other side of Eurasia, which very much resonates with our experience of modern globalization in the past 100 years. One of the key features is this geographic decoupling of the technology associated with the plant and animals versus the plant and animals itself. I think one of the things we learn from the Bronze Age is the commodities itself, cottages and livestock move in long distance without any bordering, they move very fast and across the geographical and the political borders without slowing down. But what we see is the idea and the technology associated with those plant and animals are often rejected. And certain crops was only introduced to another culture sphere when they were fully transformed within the local symbolic and the culture system. One of such example is the cooking system. So in this episodes of early globalization between 2nd and 3rd millennium BC, we see wheat and barley, for example, was introduced all the way from Southeast Asia to China. But the way in which they cooked was the Chinese way. They were transformed by the millet and rice porridge making system. And one of my students write a quite interesting paper, I think, highlighting the reduction of the grain size in East China in the context of a different cooking system. So eventually, the grain size is not only about the production mode, but also about the cooking efficiency in the context of the boiling. So if you boil porridge using wheat and barley, you prefer smaller grains and sticky starch. If you grind them for making bread, it's a different choice. And we see precisely that's a cultural choice and culinary choices in East Asia. So I think that's that's not a direct answer to the question, I'm afraid. But in a way, I think that resonates with a lot of challenges we are facing. That there's the choices of often sort of embedded in the culture and the social arrangement. And unless we can figure out some social considerations and configurations between different cultures, it's going to be difficult.
Thank you, Xingyi, very much for this.
Thank you very much. I feel that I'll travel with a Ferrari, following your thoughts.
Thank you for the question.
If I can answer to the same question from my point of view, how I experience archaeobotany in relation to what is happening today, the lessons from the prehistoric past show us how people lived with from the seeds and with the seeds for millennia, and this step to cultivation, the agriculture, has now brought the planet to a critical point. The message from the past is that people and land races survived for millennia, and it's in a way our duty to keep it going. I think that's the message that the ancestors are sending us from their charred seeds. So shall we consider our session complete? Yes, thank you very much to all the participants.
Thank you very much.